So hello grade 10 students. In this video we're going to talk about our new part for the lesson about the nervous system. This lesson is going to be about part 4 of the nervous system. Let's talk about Francois Magendie experiment. This Magendie experiment is one of the most important experiments in the field of neurophysiology. This is the most important experiment in the field of neurophysiology. So let's start step by step. This experiment includes different steps. So first of all, they did excitation of the ventral root of the spinal nerve. So there is excitation of the ventral root of the spinal nerve. We have contraction of the concerned part. Whereas the second experiment, we have the excitation of the dorsal root of the spinal nerve. You know that we have the ventral root and we have the dorsal root. And these notes, you have to know them and you have to study them. So after excitation of the dorsal root of the spinal nerve, there is contraction with the feeling of pain. So first, excitation of the ventral root of the spinal nerve, we have contraction, meaning that the ventral root, it has a motor function. Since contraction is considered as a motor function, whereas excitation of the dorsal root, we have contraction with the feeling of pain. So feeling of pain is related to sense, so it has a sensory function. Section of the ventral root. Ventral root, excitation of the central end, there is no reaction, meaning that the message stopped at this part. While excitation of the peripheral end, meaning that we have contraction, so the message continued and moves away from the center. So it moves in a way, it's called a centrifugal manner. Whereas the excitation, the sectioning of the dorsal root, and then excitation of the central end, we have contraction with the feeling of pain, meaning that the message propagates from the root toward the brain, toward the spinal cord, and then to the organ. So here it moves toward the center, toward the central nervous system. So it's called a centripetal manner. While excitation of the peripheral end, there is no reaction, meaning that the message stopped at this part. While the destruction of the spinal nerve, it shows no reaction. So we have total loss of the motor activity and sensation. Since we destroyed the spinal nerve, there is no motor activity, there is total loss of motor activity and sensation. So there is no sense and no motor activity. So this means that the spinal nerve, it has both functions. It has the sensory function and the motor one. So interpret these results to deduce the role of the dorsal and the ventral root in the spinal nerve. What did they do? First, they did excitation of the ventral root of the spinal cord. Excitation of the ventral root of the spinal nerve. We have contraction of the concerned part. So what does this indicate? This is interpret, analyze, plus indication. The indication here indicates that the nervous message was propagated from the ventral root toward the muscle. So the ventral root, it has a motor function. Whereas after the excitation of the dorsal root of the spinal cord, the concerned part, it contracts and the animals feels pain. What does this indicate? This indicates that the nervous message was propagated from the dorsal root toward the spinal cord, and then from it toward the brain, and then to the concerned part. So after excitation of the dorsal root, the concerned part contracts, and the animal feels pain. So this indicates that the message of the nervous message was propagated from the dorsal root toward the spinal cord, and from the spinal cord to the brain, and then to the concerned part or the concerned organ. So this indicates that the dorsal root has a sensory function. So first two notes, the first two important notes. First one, you have to know that the ventral root, it has a motor function. Second important note, you have to know that the dorsal root has a sensory function, and you have to study these notes. Experiment 3, they did sectioning of the ventral root, and they did excitation of the central end. So there is no reaction. So this means that the message stopped at this point, stopped at the section site. While after sectioning the ventral root and exciting the peripheral end, the muscle contracted. So the nervous message is propagated toward the muscle, to the muscle. So this indicates that the nervous message propagates in a centrifugal manner in the motor ventral root. This one you have to note, that the nervous message propagates in a centrifugal manner in the motor ventral root. It propagates away from the central, it propagates toward the periphery, since after excitation of the peripheral end and after sectioning the ventral root and exciting the peripheral end, the muscle contracts. So the nervous message propagated toward the muscle. This indicates that the nervous message propagates in a centrifugal manner in the motor ventral root. So first of all, regarding the motor ventral root or the ventral root, the motor ventral root has a motor function and the message propagates in a centrifugal manner. Whereas after sectioning the dorsal root and exciting the central end, contraction occurred and a feeling of pain was recorded. What does this indicate? This indicates that the nervous message propagated to the spinal cord, to the brain, and then to the muscle. Okay? Since we have contraction, with the feeling of pain, we have sense. Why? After sectioning the dorsal root and exciting the peripheral end, there is no reaction, meaning that the message stopped at this section and doesn't move away. Therefore, the message in the dorsal root, it moves in a centripetal manner. The nervous message propagates in a centripetal manner in the sensory dorsal root. So, what can we conclude till now is two conclusions for each root. First, for the ventral root, it has a motor function and the message propagates in centrifugal manner, while for the dorsal root, the message propagates in a centripetal manner and the dorsal root has a sensory function. The last experiment, what did they do? Imagine they destroyed the spinal nerve. There is total destruction of the spinal nerve. What happened? There is total loss of motor activity and sensation. There is no motor activity, no contraction, no feeling of pain, no sensation. So what can we conclude? So after distraction of the spinal nerve, there is no reaction and there is total loss of motor and sensory activity. This indicates that the spinal nerve has a mixed function, has both sensory function and motor function. Landline phone. Landline phone is a connection. For example, I have a phone and when I call you, landline phone is a connection between a neuron and a neuron. This connection is called synapse. But maybe it's not between a neuron and a neuron. Maybe it's between a neuron and the cell. So how does a nervous message propagate from one neuron to the following cell? We have synapse. What is synapse? Synapse is the functional connection between a neuron and other cell. This cell, maybe it's a neuron, maybe it's a gland, or maybe it's a muscle. Synapse before the neuron before the synapse is called the presynaptic neuron, meaning that presynaptic before the synapse neuron, the neuron before the synapse. After the synapse is called postsynaptic neuron. So you have to be careful with these terms. You have to pay attention to these terms. Presynaptic neuron before the neuron, and postsynaptic before the synapse, and postsynaptic right after the synapse. In the middle, we have synaptic cleft, where specific chemicals that are called neurotransmitters, they are released. Okay, so let's continue here with this synaptic density, and we have classification of synapses, either classified according to their structure or according to their function. According to their structure, we have neuronal synapse between a neuron and a neuron, neuromuscular synapse between a neuron and a muscle, neuroglandular synapse between a neuron and a gland. These neuromuscular and neuroglandular, they are called neuroeffectors, since between a neuron and an effector organ, either a muscle or a gland. So classification according to their structure, either neuronal between two neurons, or neuroeffector synapse between a neuron and an effector organ, like muscle or a gland. Neuronal synapse, they can be axodendritic between an axon and dendrite, or axosomatic between the axon and the soma, or axoaxonate between the axon and the other axon. It's called axoaxonate. So we have the axodendritic between the axon and the dendrites. Second, between the axon and the soma, it's called axosomatic, between the axon and the soma. And we have the axoaxonal, axosomatic. We have axoaxonal synapse between the axon and the axon. So how many types do we have? We have three. Either we have axodendritic between the axon and dendrite, axosomatic between the axon and the soma, axoaxonal between the axon and another axon of the second neuron. So these are according to their structure. Neuronal. Neuronal is divided into three. Either axoaxonal between the axon and the axon, axodendritic between the axon and the dendrite, axosomatic between the axon and the soma. Neuroeffector synapse. Either they are called neuromuscular synapse. We have another name. They are called motor and the plate. So neuromuscular synapse between a neuron and a muscle, it's called motor and plate. And B2 is called the neuroglandular synapse between a neuron and a gland. Neuroeffector synapses, they are between either neuromuscular synapse, which is called motor and the gland, or neuroglandular synapse between a neuron and a gland. According to their function, they are classified either excitatory or inhibitory. As their name indicates, excitatory, they cause excitation. Inhibitory, they cause inhibition of the nervous message. So, excitatory synapse, they allow or they permit the propagation of the nervous message from the presynaptic neuron toward the postsynaptic cell. Pay attention here, I didn't say postsynaptic neuron. Postsynaptic cell, maybe it's a neuron, maybe it's a muscle, maybe it's a gland. Inhibitory synapse. It inhibits or prevents or stops the propagation of the nervous message from the presynaptic neuron toward the postsynaptic cell, which is a neuron or a gland or a muscle. So as the name indicates, excitatory synapse, they have a role in excitation. They permit and they allow the propagation of the message from the pre to the post, while inhibitory synapses, they inhibit, they prevent the propagation of the nervous message from the presynaptic neuron to the postsynaptic cell. Here, the pre and the post. This is the presynaptic terminal, this is the postsynaptic. Here, the region between the two synapses, the region between two neurons is called synaptic cleft, where neurotransmitters, they are released in this region. So, neurotransmitters, they are chemical mediators, they have special roles. We will determine this in the coming slides. So, here are the presynaptic terminals. It contains vesicles, it contains sacs. These sacs contains neurons that bind to the postsynaptic neuron receptors. They bind to the postsynaptic receptors and they will cause different reactions and different chemical reactions and different, for example, effects on the human body. So as I said, this is transmission of the nerve impulse from the presynaptic neuron to the postsynaptic neuron. The neurotransmitters, the chemical mediators that are found in the synaptic vesicles, they release to the postsynaptic membrane where they bind onto their binding sites. They have special receptors. Okay? So here the nervous message is moving, then the nervous message will propagate toward the binding site, it binds, the neurotransmitter will bind and causes the Na plus, K plus bond and other ions interfere. We'll talk about them later. Propagation of nervous message. Neurotransmitter first. What is a neurotransmitter? Neurotransmitter, it's a chemical mediator between two synaptic cells. It's released by the presynaptic neuron into the synaptic cleft, as I said, where it binds to specific receptors on the membrane of the postsynaptic cell. First, the nerve impulse. This is the propagation of the nervous message through the synapse steps. First, the nerve impulse reaches the presynaptic, causing depolarization. So we have nerve impulse reach the presynaptic terminal and causes depolarization. Number two, this depolarization will open the voltage-gated Ca2 plus channels. Here we have new ion now. Last, we talk about the Ca2, K plus and Na plus. Now we are talking about Ca Ca2 plus channels. So Ca2 plus, they rush in the presynaptic buds. What do? Secretory vesicles will be mobilized, will move toward the cell membrane of the buds, and they bind to it. These secretory vesicles, they contain the neurotransmitters. So these vesicles will release the neurotransmitters where into the synaptic cleft by a process called exocytosis. Number 5, these neurotransmitters will bind to specific receptors on the postsynaptic side. First, if the synapse is excitatory, what will happen? If the synapse is excitatory, the neurotransmitter will bind to the voltage-gated or the chemically gated Na plus channels. As such, Na plus channels will open, and then Na plus will flow into the postsynaptic cell, causing the depolarization. Excitatory postsynaptic potential. It's called EPSP, excitatory postsynaptic potential. And if this depolarization exceeds the threshold, it's greater than the threshold of depolarization, so we will have an action potential. While if the steps, did you notice here the first 5 steps, they are common, but they differ starting from the 6th. If the synapse is inhibitory here, pay attention. If the synapse is inhibitory, if it's excitatory, it binds to the chemically gated Na plus channels. Here, if it's inhibitory, it will bind either to the chemically gated K plus channels or to the Cl minus. So these channels will open. So the K plus will flow out of the postsynaptic, or Cl minus will flow into the postsynaptic, causing hyperpolarization. So we are losing the positivity and we are gaining more negative here. Since the synapse is inhibitory, it doesn't allow the propagation of the nervous message. So we don't need the positive. We don't need to move to the positive like the graph, if you remember. So we need to move to the negative. So we need more negative. Either K plus, they move away from the cell, and this is a different condition that we talked before, or Cl minus will flow into the cell. Cl minus, they are anions, they are negatively charged. So this will cause hyperpolarization. It's called IPSP, inhibitory postsynaptic potential. As this synapse is inhibitory, no response will be recorded since we have hyperpolarization. There is no action potential and there is no response recorded in the postsynaptic cell. Here, I'm going to repeat this and pay attention to fix this in your mind. The first 5 steps, if you remember, they are common. First, the nerve impulse reaches the presynaptic, causing depolarization. This will lead to the opening of the Ca2 plus channels. So Ca2 plus will rush into the presynaptic buds. Then these secretory vesicles will be mobilized toward the membrane. These vesicles will release the neurotransmitter by process of exocytosis into the synaptic cleft. Also, this neurotransmitter will bind to their specific receptors where on the postsynaptic. So as a conclusion, we have a nerve impulse received at the presynaptic. This causes the opening of the Ca2 plus channels. Secretory vesicles will open, release their neurotransmitters. This neurotransmitter will be released into the synaptic cleft by the process of exocytosis. These neurotransmitters will bind to their specific receptors of the postsynaptic cell. If it's excitatory, the neurotransmitter will bind to which channels? To Na plus channels. They will open. So the Na plus will flow in the postsynaptic cell, causing depolarization. This we call it EPSP, excitatory postsynaptic potential. And this depolarization exceeds the threshold, we have an action potential. While if the synapse is inhibitory, the neurotransmitter will bind to the chemically gated K plus or Cl minus. As such, these channels will open. K plus will flow out. So we are losing the positive. Since this is inhibitory, it inhibits the positive ions to enter inside the cell. Okay, you can think like this. Inhibitory, it inhibits the entrance of positive. It allows only the negative. This will cause the decrease and the increase in the negativity. Sorry, this is called hyperpolarization, and we can divide it as inhibitory postsynaptic potential. Due to this hyperpolarization, no response will be recorded. In both synapses, in order to not to exhaust the postsynaptic, in order to let the postsynaptic terminal to relax down, this neurotransmitter will be either degraded, they are going to be degraded, and pay attention to this point. This is a very important point for the next week. This neurotransmitter will be degraded by enzymes, or they are going to be recaptured. So the presynaptic will capture them again, and let them move back to the presynaptic, or the enzymes will digest them. Okay, so this is the process here. State of convergence, we're going to talk about this in the next slide. State of convergence, it's when presynaptic neurons, they carry message to one postsynaptic cell, and the postsynaptic cell, and state of divergence, it's when one presynaptic carry nervous message to many postsynaptic cells. And this is everything for this session.